Good morning or good afternoon, everybody. I'm Estelle Leclerc. I'm an associate professor in the Department of Pharmaceutical Sciences at NDSU, and I'm the main author of this presentation. So the presentation today, I'm going to walk you through the poster, and the poster is entitled Effect of RAGE Inhibition during melanoma chemotherapy in PDX mouse model of melanoma. This study was performed in our research group, and the research team consisted of 2 graduate students, Mohamed Zahidul Hassan and Yusuf Alam, a clinician at Stanford, Daniel Tuvant, who is surgical oncologist, and my mentor for the Scholar Award, the DaCCoTA Scholar Award, who is David Bradley. So in this study, the goal of the study was to determine if the RAGE receptor participate to chemoresistance in melanoma tumor with 2 particular drugs, vemurafenib and trametinib. The RAGE receptor is a receptor for advanced glycation end product, is a receptor that has been shown in the past to participate to tumor growth and metastasis formation. And we have shown in our lab that the RAGE receptor also participates to chemoresistance when the melanoma tumor were treated with the drug dacarbazine. Dacarbazine is a DNA alkylating agent. If we look at figure 1, the first figure of the poster, it demonstrates, it shows the molecular pathway which leads to chemoresistance through the activation of RAGE. Studies have shown that when melanoma tumor are treated with dacarbazine, there is a release in the tumor microenvironment of different molecules which are called alarmin. 2 of these alarmins are HMGB1 and S100B. There's also alarmin S100A4, S100A6, S100A8 or A9. These different molecules are ligand for the RAGE receptor. RAGE become activated, and once RAGE is activated, it leads to the activation of downstream signaling pathway. And one of these pathway is a MEK-ERK NF-kappa B pathway. Through the activation of this particular signaling pathway, this leads to the growth of melanoma tumor despite the treatment with a cytotoxic drug, and this leads to chemoresistance. One particular feature or property of RAGE is that when RAGE is activated, this leads to the upregulation of RAGE because RAGE is under the expression of RAGE is under the control of NF-kappa B. So this leads to an amplification of the RAGE signaling. So we have shown this in the past with the cytotoxic drug dacarbazine, and we wanted to ask the question in this new study if RAGE activation also leads to chemoresistance when the melanoma tumor are treated with vemurafenib and trametinib. Vemurafenib and trametinib are the current standard of care for melanoma treatment. Dacarbazine was the standard of care for melanoma treatment until about 10 years ago. But now, one of the standard of care for the treatment of melanoma is vemurafenib and trametinib. And I need to mention that vemurafenib only is able to treat melanoma tumor only if the tumor, they carry a mutation of this particular kinase, the BRAF kinase. So how do we want to demonstrate, how did we want to demonstrate this, the role of RAGE in chemoresistance? So we decided to demonstrate this effect in vivo using patient-derived xenograft tumor. So this patient-derived xenograft mouse model of melanoma tumor, they are generated from tumor from melanoma patient. And through our collaboration with Sanford and Dr. Daniel Juvent, with an approved IRB, we were able to obtain 5 different melanoma tumor from different patients. From these 5 different melanoma tumor sample, we were able to successfully implant and expand 3 of the 5 tumor samples. The 2 other tumor samples were not able to be expanded in mice. So we were happy to be able to have a 60% success rate in the generation of our melanoma PDX. With this expanded tumor, before we move on to test how the effect of RAGE inhibitor on this tumor, it was very important to determine if the tumor we obtained from patients were sensitive to the drug vemurafenib, and whether, because this is a condition in our study. So to do that, we decided to test the sensitivity of cells deriving from the tumor for vemurafenib. For each of the tumors that we implanted in mice, we generated a cell line. So we use this cell line and expose the cell lines to different concentration on vemurafenib, and this is what we have in figure 2 in the poster. In figure 2, you can see a bar graph which shows the metabolic activity of the cells deriving from the PDX tumor to 2 concentration of vemurafenib, 0.41 micromolar of vemurafenib and 11.1 micromolar of vemurafenib. And we've shown that there is a dose-dependent response of the cells toward vemurafenib with IC50 of about 0.41 micromolar of, or 0.5 micromolar of vemurafenib, which shows that at least one of the tumor we expanded is sensitive to vemurafenib, and we will be able to perform the next step of a study with at least this tumor. It was also very important to show that the cells that we derived from the tumor were melanoma cells, and for this we did the immunofluorescence staining using a marker of melanoma, which is melanMART1, and this is shown in figure 3 in the poster. So the cells staining positive for melanMART1, the marker of melanoma, are stained green, and the nuclei of the cell are stained blue using DAPI. So we were able to show that at least for one tumor, the cells are melanoma cells that we tested, and they are sensitive to vemurafenib. So with this, we will be able to go to the second step of our study, where we want to determine if RAGE participate to chemoresistance with the 2 drugs, vemurafenib and trametinib. In this study, we will use the PDX mouse model. We will use mice carrying tumor expanded from the patient sample, and we will have several groups of mice, but the 2 important groups are going to be one group, the mice will be treated with a combination of vemurafenib and trametinib, and another group, the mice will be treated with a combination of vemurafenib, trametinib, and the RAGE inhibitor. And we anticipate that in the presence of the RAGE inhibitor, the tumor are going to be smaller than in the absence of the RAGE inhibitor. So with this concludes the presentation of this poster. I want, as a last sentence, I want to thank the sponsor for this work, which is in part the NIH with the DaCCoTA Scholar Award, and as well as NSF North Dakota EPSCoR. Thank you for your attention.